Good morning, Biology 103. Today we're going to be talking about cells. So, the cell is just the basic unit of life. If we zoom into any living thing, anything that we would categorize as living is going to be made of at least one cell. So, cell theory, it's a combination of multiple different discoveries from different scientists all put together. Some came from observations of plants, others from observations of animals. But essentially, the cell theory is just that all organisms are made of cells. These cells are the basic units of structure and function. And to get cells, you have to have cells. So cells come only from other cells. Cells have the same basic composition, use energy, and contain DNA. No matter what we're talking about, if we're talking about living things, it's going to meet this criteria. There's a few different scientists who played a major role in, first off, not even just cell theory, but just figuring out that cells were even a thing. And Robert Hooke was one of the first people to do this. In 1665, he discovered the cell. And if you look at the cells, they look like little prison cells. And it's named after that for the compartments that he was able to actually observe. He used a really kind of complicated for the time microscope, but it's really simple. It just has a few lenses in sequence. There would be a lens in here to magnify the image. And I want to say it was two convex lenses that were used. It might have been three, but again, convex would just mean it has curved glass rather than it's bowing out rather than convex, where it kind of bows inward. The image you would get from this microscope would be terrible. You could barely tell what it was. So what Hooke ended up doing, because really, and you know when you use those microscopes in the lab, that the farther you zoom in, the darker the image gets. And this was a particular problem with this type of microscope. So what he developed was this little water flask, a light source, and then of course it had to have a fuel, fueling the flame. And what this was allowed him to do was to have extra light coming into the subject so he could actually see it better when it came through the microscope. Overall, though, not a very good microscope. And you would think that it would be decent compared to Leeuwenhoek's, but actually Leeuwenhoek's was simpler and better. Now, this is one of his drawings, so even though his image was super dim and hard to see, with his modifications, he was able to get pretty detailed drawings. Then we get to Leeuwenhoek, and this is a couple, about 15, 20 years later, and he was the first to see cells that were like living, active, and he saw these in pond water. And this was his microscope. You can see it's much more simple. It's just got one little lens there, and then it had this little sample holder that you could just prick down. Maybe you have a leaf or something you want to look at. You could just prick it on the end of that needle, and then you could adjust this knob, which would move it closer or farther away from the lens to increase the focus if you're trying to get the image to be sharper, because it's going to look blurry at first. And then this right here, the translator would allow him to move that image up and down. And Leeuwenhoek's microscope, even though it looks much simpler, if you just saw it, you might think it was a precursor to Hooke's. It was actually better, and he was able to get more detailed drawings from his microscope. And that brings us to Sleiden, and he was in 1838, so now we're jumping almost 200 years later. And he observed that all plants, every plant he observed, was made of these little cells. And we also had the same observation with animals. So then Schwann comes in, and he's like, well, every animal I look at, I zoom in close, and I'm looking at cells. And you look at the year here, it's the same time period. And they were actually able to work together on this and contributed to the cell theory that all living things are made of cells. And then Rudolf Virchow in 1855, he had this famous saying, which was just to say that all cells come from other cells. So he was able to see this propagation taking place. No one has ever seen, nor will they ever see, cells come from anything other than cells. Life comes from life. And then we get to Janet. I'm not sure how to say her last name. I always just say Plow, but maybe it's Plow. I don't know. But anyway, in the early 1930s, she determined that the cell membrane was a physical structure. Because if you look at cells under the microscope, and I'll see if I have, well, you can kind of see it there in the bottom. Not so much, though, from that picture as you can, like when you're really looking under your microscope, what it looks like. But they all seem to just kind of blur together. And there was a debate as to whether or not that cell membrane was actually something physical, or if it was just where two different liquids might be coming into contact with one another. You know, sort of like when water touches oil, it looks like there's a layer there. And you might think that there's a structure, but it's actually just two different liquids coming in contact with each other. And that was one of the big debates, and she showed that that membrane was actually something physical. So then we get into the microscope, and this is what we'll be using in the lab as we go forward, hopefully a good bit. But it just shows you what are some things you can see without a microscope. Well, you might be able to see an egg, you might see some frog eggs, maybe even a human egg. But when you get to this point, when you start talking about cells, is where you really need microscopes to really tell what you're looking at. And look, just from a light microscope, you can see all the way down to these really tiny bacteria. If you take a microbiology class, you'll be using the same compound light microscopes that we use in biology, and you're going to be able to see through a gram stain extremely small Staphylococcus bacteria, and you'll see it in pretty good detail. Now, when we talk about electron microscopes, you can get down to literally, you can see the fundamental component of all matter. You can actually get down and look at atoms. And they don't show it showing that right here, but they actually do have electron microscopic images of atoms, and they just look like blurry clouds, like a gold atom you could Google right now, and it just looks like a blurry mass. So what are the relative size of some of these things that you might observe under electron microscope or a light microscope? Well, I think it helps because we've all seen just a single hair. You've picked one up, you can see it, but it's insanely thin. So if we use that as kind of our standard of comparison, well, you've seen a grain of sand from the beach. That's actually could be, if it's a single piece, it would actually be thinner than a hair. A grain of salt could get even smaller. And then we look at a white blood cell. So you might actually be able to see that with the naked eye. It would be, because white blood cells can be pretty large because they engulf, bacteria. You look down here at bacteria. These guys are going to be eating these guys. Or these white blood cells will be eating these guys. But the farther down you go, you really need technology to be able to observe these. So you see a red blood cell is much smaller than that. And just think about this. We are all familiar with red blood cells in comparison to a single strand of hair. Look how much smaller that is. So tiny. And then it keeps going and going and going. You got this little bitty Zika virus all the way down there. And then you've got this little T4 bacteriophage, which is a virus that infects bacteria. So when we talk about cells, one of the questions is, well, why are they so darn tiny, right? Why don't we just exist as one huge blob of cell that just absorbs nutrients and releases waste? Well, the issue is when you get a really big cell, they're really hard to feed. And we'll talk about transport. Cells need to be able to move things in and move waste out. Maybe even move hormones out or signaling molecules. And those things need to happen quickly and efficiently. And when you get a huge cell, you have to move those particles so far before they're able to escape the cell, especially if they're produced maybe near the nucleus somewhere. So really it's beneficial to have a small cell where those molecules can be shipped out faster. And it's not just a distance thing, how far they need to go. It also has to do with how much surface area the cells have in comparison to their volume. You'll see this little chart here. It has surface area to volume ratios for a huge cell. In this case, we're using cubes as examples because it's easy to calculate the surface area and volume. And then for medium cells, and then this tiny little cell. If you look at the surface area and the volume, you'll see how they're both decreasing as you get to a smaller cell. But if you look at the ratio, like right here with this large cell, the surface area is smaller than the volume for the really big cell. But then as you move down, now they're equal. Now the surface area is larger than the volume. And then you get to this really tiny cell, and the surface area is 3 times as large as the volume. So the key to a cell is to have a high surface area to volume ratio. And this cell that has a surface area to volume ratio of 3 is much better, more efficient than this cell that has a surface area of 0.75, not to mention that that cell is going to need way more nutrients to even feed all of its tissue. And it's going to be way harder to move waste out and bring nutrients in. So the idea is the smaller the cell, the higher the surface area to volume ratio, which means it's easier to move materials in and out very quickly and efficiently. And when we talk about what are cells even made of, there's basically 2 parts. They have a cell membrane. There's something that separates them from the outside environment. And then they have everything on the inside, which we call the cytoplasm. It's a sum of all the interior components of the cell. We think about cytoplasm, we think of the gooey substance on the inside, but that's actually the cytosol. Cytoplasm includes all of those things on the inside. So that's the basic structure of a cell, but what kinds of cells are there? And it depends on how specific we're going with that. But if we're talking about generally, what are the types of cells? There's only 2 types. You have those that lack a nucleus, which we call prokaryotes. They also lack organelles. And then we have those cells that have a nucleus, which we call eukaryotes. And that word eukaryote actually means with a kernel, like a kernel of corn, because when they looked at these, they had this little like corn kernel looking structure, which was the nucleus, which they didn't know what it was, but they knew it had something that these other cells did not have. Prokaryotes have a nucleoid region, which is where the DNA is, but it's not enclosed in a membrane. Eukaryotic cells have a nucleus enclosed in a nuclear membrane and has all these organelles enclosed in membranes. So when we talk about prokaryotic cells, they do not have internal membranes. They lack all the organelles. Most importantly, they lack a nucleus, which comes back to the name of prokaryotes. And the nucleus is membrane-bound DNA when we're talking about eukaryotes. Prokaryotic cells have DNA, but they are missing the membrane covering. All prokaryotic cells are bacteria. So all of our bacteria are prokaryotes. Talk about eukaryotic cells. Just by looking at it, you can tell they're much more complex. Usually going to be larger because they have a lot more in them. Just to house all that extra DNA would require a lot more space. Like the nucleus of a eukaryotic cell is probably going to be significantly larger than the entire cell of a prokaryote. Prokaryotes are super tiny. They have internal membranes around those internal organelles, like the mitochondria, the Golgi, the rough and smooth endoplasmic reticulums. Rough and smooth. And all the structures we'll get into. And there's different subtypes of eukaryotic cells. As we talk about animal, plant, protists, and fungi, which we'll get to all of these eventually. So what are some of those organelles in the cell? Well, the cell membrane isn't an organelle, but it's a huge structure that covers the entire cell. And even those organelles have membranes around them. Ribosomes, these are these protein factories, or these machines that produce proteins. Those are made in the nucleolus, within the nucleus. Then you have the cytosol, that's that liquid substance that the organelles are in. And then, of course, these cells have DNA and chromatin. And this is what's common to prokaryotes and eukaryotes. They all have a cell membrane, ribosomes, a substance that fills them, cytosol, and they're going to have DNA. So that means all living things have these in common, whether they're prokaryotes or eukaryotes. But when we talk about eukaryotic cells, we have to differentiate between what's just such a common topic within biology, and that's plant versus animal. Hopefully you're already pretty familiar with this. But there are, of course, 4 types. But we're going to talk about plants and animals. So again, if we're talking about eukaryotic cells, which is what these would fall under, they're going to have a lot of things in common. Both going to have a nucleus. They're both going to have an ER, ribosomes, Golgi, membrane, and cytoplasm. But if we're talking about a plant cell, they have a few extra apparatuses. They have chloroplast, a cell wall made of cellulose, and then they have these central storage vacuoles for storing water. And these are going to give them a few different characteristics. Of course, they can do photosynthesis, and then plant cells can store a lot of water in those vacuoles, which can help them use that water later when they need it. For photosynthesis, it also gives them their shape and their structure. And then the cell wall gives them a lot of strength and rigidity so that they can grow to be massive multicellular structures like we see with, the redwoods, perhaps. Animal cells have their own unique structures. So they have these centrioles that aid in cell division. We talk about mitosis, meiosis. We'll talk a lot about those. They have these cilia, which are these short hair-like structures composed of this 9+2 arrangement, which we'll look at a picture of that. And cilia can help with movement, of course. You've probably heard of a flagellum before that allows animal cells to move quickly. We have these basal bodies, structures that anchor the cilia and flagella to the cell membrane. And then lysosomes. These are these digestive sacs that can break down things that are in the cell and also could help a cell move through cell death, or what's called apoptosis. So here's just a couple images of flagella and cilia. Now cilia, we think about the lungs. This would be more for moving things across the surface of the cell. Could be for movement. Now flagella are going to be for like really quick movement. But cilia helps like moving things around the cell. So in our lungs, it can help us move mucus out of our lungs so we can cough it up. And these are those basal bodies and some cilia. I believe this is actually a flagellum. But you see this 1, 2, 3, 4, 5, 6, 7, 8, 9 plus 2. So that 9 plus 2 arrangement. That's just their structure of microtubules. So then what are the unique structures to the plant? Well, of course, they have the chloroplast, which is full of chlorophyll pigment, and that's where photosynthesis is going to occur, and that's where glucose and sugar will be made. And we'll spend a lot of time talking about that when we get there. They have leucoplast, which stores starch. Think about like potatoes. And then chromoplast, which store pigments. So these are collectively called plastids and are sometimes called organelles of color. So they can give plants their color. And you'll see with plant cells, they're often depicted as like hexagonal or like rectangular. It's because these cell walls give them a lot more structure versus animal cells. They're usually a little more flexible. And then you'll see here we got the cell wall, which is made of cellulose. So it's literally made of sugar, and it's because of the arrangement that it's in, it can be extremely strong. You got middle lamellae. This is the region between plant cells that cement cells together. Especially with plant cells, you really want them to be able to connect well because they have to hold so much weight, and they're trying to grow up competing with nearby plants to get sunlight. And then they have these vacuoles that help them store water and food for use, and it helps them maintain their shape. So here are some organelles that animal cells and plant cells have in common. And we'll talk about all of these. So the nucleus, hopefully if you've taken a biology class before, all of this is just boring and familiar to you. But the nucleus, they call it the control center, and it controls what happens in the cell because that DNA is what's going to regulate, what can be made in the cell, what proteins can be made or ultimately coded for in the DNA. And those things are going to be running the operations of the cell. So the nucleus, what DNA is in there, is going to control what happens, and that's what's going to be passed down to the next cells that are made from it. So it's hereditary. The nucleolus is that center of the nucleus of the nucleus, and that's where ribosomes are built, and ribosomes build proteins. And then you have the nucleoplasm, which sounds like cytoplasm, and that's the cytosol within the nucleus. You've got the cytoskeleton. This has these 3 different types of fibers or rods that give the cell its shape. Cytoskeleton, keyword there is skeleton. Cyto means cell. It's the cell skeleton. It's the skeleton that holds our cells in their shape, just like our skeletons hold our bodies in our shape. There's 3 types of filaments. Micro, which are the smallest. You see a lot of these in muscle tissue. And then you have microtubules, which are larger and hollow, that can be sort of function like railroad tracks, and we can move things around in our cells as they're anchored to these microtubule tracks, and they can be moved down and up from these microtubules. And then you have the intermediate, which are in the middle as far as size goes, and they provide internal scaffolding and help create cell shape. The endoplasmic reticulum, you have smooth and rough. I don't think I put, let's see. Yeah, this is supposed to say rough right here. But anyway, the rough ER, they call it that because it looks rough. If you look at it, it has all these little bumps on it, and those little bumps are ribosomes. So this is where proteins can be made that will then be put into this transport system to be moved outside of the cell. And then you have the smooth ER, which is going to play a role in detoxification. So you see a lot of these in our liver. Then you've got the Golgi apparatus. I call it the FedEx. That's where proteins can be packaged, modified, and then be shipped outside of the cell. You've got basic vesicles. These are just a membranous sac that can store things. Peroxisomes, vesicles containing enzymes that neutralizes hydrogen peroxide. Mitochondria, if you've heard of cellular respiration, if you haven't, you're going to hear a lot more about it than you want to. But that's where energy is going to be made, and the key molecule there is going to be ATP. That's what we have to get anything done in our cells. So we have to be able to make that somewhere, and the mitochondria provides that for us using glucose and oxygen. Then we've got vacuoles. These store food, water, waste. Plants contain a large central vacuole. In animal cells, they can still have vacuoles, but they're going to be a lot smaller because they don't need to be as large. And here's just a picture of an animal cell. So just being familiar with the basic structure of plant and animal cells, what are those organelles in each, and what do they do, is what we're hoping to understand by the end of this section. If we're talking about prokaryotes, remember, these are those simple cells. They don't have the organelles. They don't have a nucleus. They do have some cool structures, and one of those is a pilus. And this can allow them to connect to surfaces, which can make bacteria stick around maybe in our lungs longer than we want them to. And it gives them an advantage because they're able to stay in an environment and get nutrients and food for longer than they would without those. They also can produce a capsule, which can make them sticky. It's the slime, like mucus-looking covering, which can make them extra hard to get rid of, harder for drugs to come in contact with them, and harder for our bodies and immune systems to fight them off. So these structures give them a lot of strength and make them harder to kill. And then they have the nucleoid region, which is where the DNA is going to be located. It sort of stays in this one spot, and we call that the nucleoid region, even though it doesn't have a nucleus. The next section is just more cellular structures. You're welcome to look at those if you want to. They'll be on Canvas.